The overall goal of the presented modified chick embryo model is to characterize substances with insulin-mimetic properties in a living organism.
This method can help answer key questions in the field of diabetes research, such as finding herbal extracts with insulin-mimetic properties that reduce blood glucose levels without side effects. The main advantage of this technique is that the efficacy of compounds can be tested in a living organism with adequate throughput rates, and that permission by an ethics committee is not necessary. Visual demonstration of this method is critical, as the preparation and cutting steps are difficult to learn, because the vessels are not easily prepared or often cut completely and get lost.
Check eggs for fertilization after incubation at 38 degrees Celsius with an average humidity of 40 to 60% for up to 11 days. To do this, dip the fringe of the candling lamp into an ink pad, then candle the pointed side of the egg. To identify the air bladder, check for differences in brightness on the top of the egg. The air bladder appears as a round, more transparent region, while the albumin appears darker. Mark the location of the air bladder by pressing the lamp slightly against the egg with the previously applied ink. Exclude non-fertilized eggs which do not show a difference in brightness between the albumin and air bladder. Prepare the insulin mimetic or insulin by diluting to the desired concentration in Hank's balanced salt solution. To apply this